In this video, we're going to be looking at the elongation stage of transcription. Remember to like the video and subscribe for more high-quality medical education content. Just to remind ourselves, transcription consists of three main stages. So firstly, we've got our initiation stage, which we talked about in a previous video. And then we've got the elongation stage, which we're looking at here. And lastly, we've got the termination stage, which we'll look at in a future video. Now, in this diagram, we've got a few different things going on. And we've got our DNA strand here. And specifically, we're looking at the template strand, which is implicated in transcription, because copying this is going to result in our messenger RNA molecule. Now, specifically at this stage, we would call it pre-mRNA because it undergoes a lot of post-transcriptional processing before it becomes a final messenger RNA molecule. And what else have we got? Well, we've got our ribonucleotides here. And we can see that these guys are entering a molecule, which we'll remember from a previous video. It's called RNA Polymerase 2. Specifically, they're entering a part of the molecule called the entry site. That's the entry site. And what we're going to do is just move on to the bottom right-hand diagram, just to look at this in a bit more detail. So we've got our DNA strand here, and then we've got our messenger RNA here. Now, in our DNA strand, we've got 3 nucleotides shown. Why is this relevant? Well, 3 nucleotides in DNA is known as a base triplet. That's a base triplet. And when we've got our mRNA molecule being formed in transcription, the corresponding ribonucleotides to a base triplet is called a codon. So that's 3 ribonucleotides which correspond to a base triplet. Okay, so now back to the top diagram. The ribonucleotides are moving into the entry site, and when the right ribonucleotide is identified to join on to the corresponding nucleotide at that point, the new ribonucleotide moves into the active site. That's the active site. And then joins on to the messenger RNA molecule being formed. Now, how does it do that? Let's take a look at the diagram on the bottom left-hand side this time. You've got these things here called phosphate radicals, right? That's phosphate radicals. We've got 2 of them specifically. And of course, we've got our nucleotides, or our ribonucleotides here as well. Now, what actually happens is the RNA polymerase 2 molecule, before transcription takes place, it actually adds on these phosphate radicals onto the ribonucleotides to activate them for the process of transcription. And what's happening at the elongation stage is essentially the exact opposite. So the phosphate radicals are coming off, and this process is producing a lot of ATP, which is the energy source required for forming phosphodiester bonds to link the ribonucleotides onto the rest of the messenger RNA molecule. And we'll remember that the messenger RNA molecule is being formed from the 5' end to the 3' end, whereas the template strand of DNA goes from the 3' end to the 5' end. So that's a quick overview of the elongation stage of transcription.